Our bones are a marvel of natural design, continuously rejuvenating through a finely balanced equilibrium of bone resorption and new bone formation. A truly miraculous structure is formed which follows the lines of mechanical load, the trabecular bone. Sometimes, however, the unexpected happens. A drill intrudes and destroys the naturally sculpted bone. Blood vessels are torn and a major defect is created, quickly filling with blood. The titanium implant is inserted where the tooth was previously lost. At first, the only force holding the implant in place is mechanical friction. This is called primary implant stability. Osseointegration, or secondary implant stability, requires a highly complex sequence of additional biodynamic processes. This is facilitated by finely tuned communication between the main actors of wound healing, the cells. The blood immediately perfuses the surgical site, providing the cues for subsequent healing. Within seconds or minutes, ions and serum proteins such as albumin, fibrinogen, and fibronectin begin adhering to the titanium surface. Next, the bleeding is stopped by blood platelets, also known as thrombocytes. When they are exposed to collagen and other proteins from the traumatized tissue and implant surface, they aggregate and close the ruptured blood vessel. Platelets release various messenger substances for cell-to-cell communication, such as thromboxane, which promotes platelet aggregation, or PDGF, platelet-derived growth factor, which stimulates the cell division of fibroblasts. Fibrin monomers spontaneously cross-link, forming a fibrin network. The blood clot permeates the wound space, forming a provisional matrix. It also adheres to the implant surface. This blood clot has tremendous importance as a provisional matrix for subsequent bone healing processes on the implant surface. During the early stages of healing, immune cells clean the wound of the very fine bone chips, tissue debris, and oral bacteria that remain following the surgical procedure. In a first step, bradykinin from the platelets increases blood vessels' permeability. As a result, the endothelial cells move apart very slightly. On the inside of the vascular walls, endothelial cells promote the attachment of polymorphonuclear leukocytes from the bloodstream. These leukocytes, also known as PMLs, squeeze themselves through the gaps between the endothelial cells. Once they have digested the basal lamina using proteases, they are free to enter the wound. PMLs chemotactically navigate toward the wound along a molecular concentration gradient. These molecules include bacterial proteins, fibrinopeptides, and pro-inflammatory interleukins. Upon arrival, they kill bacteria through the release of reactive oxygen species. PMLs also release highly digestive enzymes such as collagenase and elastase. The wound then proceeds through an uneventful healing process, or a toxic wound environment develops with elevated bacterial counts and toxic byproducts, potentially leading to wound breakdown and implant loss. PMLs can call for